Hi everyone, welcome to our channel Math Tonic. And I know it's a very crucial time for you since you have your exam on Tuesday. And I have made a predicted paper for the same for the May June 2025 paper. But just a piece of advice, please be focused in reading the questions, do them properly. You have all the concepts with you, you have worked really hard. So stay focused while reading the question. Most of the time, you make mistake in that. And stay calm, answer as per the marks. Okay. So, without further wasting time, let's start. Okay. So, starting from question number 1, figure 1.1 shows a diagram of human skin in hot weather. Name structures A to D. Okay, so A is basically here. So, A here is your sensory neuron. Okay, B is your thermoreceptor. Okay, C is your sweat glands. Okay, and D is your fat tissue. So this labeling you have to do, and for this you will be awarded 4 marks. B part. Describe how the structures A to D in the skin help to maintain a constant body temperature. You may refer to the structures by their letters. So either we can use letter, either we can use the terms also. So first of all, you have to describe how the skin will maintain the temperature. So we can take two ways. Either there is increase in temperature or decrease in temperature. So here I have taken increase in temperature. Increase in temperature is detected by thermoreceptor. Thermoreceptor is your B, which is present here. Okay, so thermoreceptor B. They send information to hypothalamus using A. Hypothalamus sends impulses to C, and sweat production to produce cooling effect. So basically, they send information to hypothalamus using A. Now A here would be your sensory neuron. Okay, and then from there information will go to hypothalamus and then it will come to C. So C is your sweat glands. So sweat glands will produce sweat and this will produce a cooling effect. So C, sweat production to produce cooling effect on the evaporation. Okay? So when the sweat will evaporate, it will produce a cooling effect. Hair erector muscles will relax to lie flat and trap no air, so that there is no insulation and basically you will have no air trapped, so that the body can cool down easily. So this is basically your B part. CI, blood flow through the skin changes in response to the changes in the air temperature. State and explain what happens to the blood flow through the skin when the temperature of the surrounding air becomes cold. So here we are going to describe how your skin is going to play a role in you can say bringing your temperature back to normal when you're feeling cold. Okay, so this is, I'm going to start with vasoconstriction of the arteriole. Vasoconstriction of the arteriole means the arterioles which are supplying blood to your skin capillaries, those will actually reduce in diameter so that less blood is supplied to your skin. And moreover, whatever blood will be passing through the through this arteriole, from that more blood is going to flow through the shunt vessel. So overall, the blood circulation to the skin capillaries will be reduced. So the blood flow to the skin capillary is reduced. Now how is this going to help? Explain how the changes you have described in CI reduce the heat losses. So what is happening here is we are reducing the overall blood supply to the skin. So when less blood will actually flow to the skin capillaries, less heat will be lost from the blood by radiation. So this is how we are going to reduce the overall heat losses, because as it is your body is feeling cold, there is a drop in temperature. So along with other activities which will be going on in the body to increase the temperature, this is one way to reduce the heat losses by reducing the blood flow to the skin capillaries. Okay, moving ahead to D part. The control of blood glucose is an example of negative feedback. Explain how negative feedback controls the blood glucose concentration of a person who has not eaten for a day. So you have to describe how negative feedback is going to help and bring back the temperature bring back the blood glucose level back to normal if you have not had a meal in a long time. So first of all, because you did not have any food, so normally the blood glucose level will actually fall below the normal, which is detected by pancreas. It has receptors, it will detect that, and pancreas secretes the glucagon. So pancreas here secretes glucagon, Glucagon acts on liver cells to break down glycogen to glucose. So pancreas is going to release the hormone glucagon. Glucagon is going to act on the liver cells, and the liver cells will break down glycogen to glucose. This glucose will be released in the blood, and the blood glucose concentration will be restored or brought back to normal. Coming to question number 2. Figure shows a fetus in the uterus immediately before birth. Describe the functions of the amniotic sac and amniotic fluid. Now you can write, this question is of 4 marks, so you can write 4 points related to amniotic sac and amniotic fluid. So whatever points I have covered here, I will tell you, and I might tell you 1 or more points extra. So amniotic sac helps to maintain the temperature of the body. Amniotic sac acts as a cushion to protect the babies from mechanical injuries. So it's like a cushion, it's like a sac which basically protects them from any injury. It's like a sac to maintain the body temperature. Amniotic fluid protects the pathogen from reaching the baby. Yes, it acts as a barrier to protect the babies from any kind of infection, from any kind of pathogens. And amniotic fluid allows the body to move. It also allows the baby to grow properly, because it's a sac filled with water, so basically it will allow the baby to move easily, to grow easily. So this is how the amniotic sac and amniotic fluid help in the growth of the baby. Amniotic sac, amniotic fluid also help the baby in excretion. So this is also one more point which you can light. List 3 functions of placenta. 3 functions of placenta. So 3 important functions of placenta that I have mentioned here is: placenta supplies nutrients like glucose, amino acids to the baby. Placenta removes waste like CO2 and urea out of the baby's blood. And placenta secretes hormones like estrogen during pregnancy. So these are the main functions of placenta. Discuss 2 of the advantages of breastfeeding. Breast milk provides balanced nutrition for the growth of the baby. It has antibodies to provide passive immunity to baby against infection. So it gives time for the baby's immune system to develop, as well as it has a balanced amount of nutrients for the baby to actually grow. Coming to the D part. So in D part, the question is: outline the events that happen after insemination till implantation. So these basically what you have to do is, since the sperms are actually deposited in the female reproductive tract, after that till implantation you have to go through the entire process. Okay, let's just see how I've written the answer. Sperms travel to the oviduct. Sperm fuses with egg cell. Haploid nuclei of the egg and sperm fuse to form a diploid zygote called fertilization. Zygote divides by mitosis to form ball of identical cells called embryo, and this embryo now implants in the wall of uterus by placenta. Please keep this in mind while you're writing about mitosis, while you're writing about zygote. You always have to write that they are a ball of identical cells, or mitosis produces identical cell. This is short a point every time when you mention mitosis. Okay, and for zygote you can also write diploid zygote. Sometimes in the marking scheme there is a mark separately mentioned for diploid and for sperm and egg cell to be haploid. So these points, when you're writing these keywords, definitely you are aiming on a high score. Moving ahead to question number 3. Penicillium is an antibiotic produced by the fungus Penicillium chrysogenum. So for your information, I'm telling you, only thing that you need to remember is penicillin is produced by a fungus. The name of that, the genus of that fungus is Penicillium. You don't need to remember any specific species, but you actually need to know that Penicillium is the fungus which produces penicillin. First part. Enzymes in the fungus are used to make penicillin. Explain why there is a water jacket around the fermenter and nutrients are added to the fermenter. So 2 particular points you have to explain. What is the need of the water jacket and what is the need of the nutrients. Okay. So here water jacket here is present here, over here, and this water jacket actually helps to maintain the temperature of the fermenter. Okay, So just see the answer. Heat is produced as a result of aerobic respiration. Water jacket helps to remove the extra heat to maintain the temperature and prevent the enzyme from getting denatured. Another point which you can add here is enzyme need an optimum temperature to function. So this is how you can write, but this is enough to get marks. 3 marks. Nutrients. Glucose to undergo aerobic respiration and release energy for growth of the fungus. Second thing is you need amino acids for actually making proteins and enzymes of the fungus. Okay. Then figure 4.2 shows the mass of the fungus and the yield of penicillium during the process. So this is the graph representing how the penicillium was produced by penicillin was produced by penicillium. Now BI, state the time interval over which the fungus grew at the maximum rate. So this is the particular part where the fungus actually grew the most, and that's what we have to write here. So this is exactly representing the growth of the fungus, and this was the point where it actually grew the most. So I have written it 40 to 50 hours. As the fungus grows in the fermenter, the nuclei and the fungus hyphae divide. Say the type of nuclear division that occurs during the growth of the fungus in fermenter. It is mitosis. Please make sure and please be clear. The type of division that we are talking here is nuclear division. Nuclear division, mitosis and meiosis. So meiosis will produce haploid gametes and mitosis will produce your diploid to diploid cell, which are identical. III. Explain why the growth of the fungus slows down and stops. So here, why the growth of the fungus actually slows down is because of the limiting factor. The lack of nutrients of glucose builds up of toxic waste, lack of oxygen supply and no space to grow. So this is because when the fungus stops growing is maybe too much of waste is accumulated, maybe the nutrients are not properly present, maybe the oxygen supply is limited, or maybe there is no basic space for the fungus to grow. So these all points you can include in your answer. Question number 4. Figure 4.1 is a diagram of Vibrio cholerae, the bacterium that causes cholera. Okay. Explain how cholera bacterium causes diarrhea. So just let me brief you the process. Cholera bacterium releases toxin which causes chloride ions to move into the lumen of the small intestine. When the cholera toxin is going to be released, it causes the chloride ions to move into the lumen. When the chloride ions they come in the lumen, they reduce the water potential. As a result, water from the cells moves into the lumen from higher to low water potential by osmosis, and this results in diarrhea, dehydration and watery feces. B. A scientist tested the resistance of one strain of bacteria to different antibiotics. The scientist tested solutions of 5 different antibiotics A to E. She soaked a paper disc in each antibiotic solution. The paper disc with antibiotics were placed in a petri dish containing bacteria on agar jelly. Figure 4.2 is a diagram of appearance of the petri dish after 48 hours. The shaded areas show where bacteria grew. The clear areas show where bacteria did not grow. Okay, so A, B, C, D, E. What do you think is the most effective antibiotic here? The most effective antibiotic here is E. Why is it the most effective antibiotic? Because it shows the more clear area, more zone of inhibition where the bacteria could not grow. Moving ahead to first part. The strain of bacteria used in this investigation causes a disease. Using the information in figure 4.2, explain why antibiotic E would be most effective at treating this disease. So as I just told you, because there is most area, clear area where the bacteria could not grow, most zone of inhibition, that is why it is most effective. I. The results in figure 4.2 show that this strain of bacteria is resistant to antibiotic A. Let's just scroll up. Okay, so this is bacteria A, and this bacteria A, they are saying that this this antibiotic A, the results in figure 4.2 show that this strain of bacteria is resistant to antibiotic A. 5 years ago, a similar investigation was found that the clear area for antibiotic A was the same size as antibiotic B is in figure 4.2. Explain how bacteria becomes resistant to antibiotic. So let's just show you how it is. So this is A. You can see the clear zone is like more or less not present, and here you can see B, where the clear zone is very big. So they are saying that A also had a clear zone, but now it has become you can say the bacteria has become resistant to this antibiotic A. Okay, explain how bacteria becomes resistant to antibiotic. So some bacteria have resistant gene because of mutation. So you can understand that any particular new gene is going to develop because of mutation. So the resistant gene here also, it develops because of mutation, but this difference in resistant and non-resistant bacteria is only going to show up when we use the antibiotic. So when antibiotic is used, it causes the resistant bacteria to survive and the non-resistant bacteria to die due to natural selection. Resistant bacteria reproduce with competition and increase resistant alleles in population. So when the resistant bacteria, they basically when antibiotic is used, the resistant bacteria, they survive, non-resistant, they die. The resistant bacteria now reproduce because there is no competition, and they increase their number as well as their alleles in the population. Third part, describe how to minimize the risk of antibiotic B developing the same results as antibiotic A. So how to minimize the risk? By taking correct dose and not overusing it. First thing is taking the proper course of antibiotic, that is taking proper dose, and second thing is not taking it again and again, not overusing it. So these are two separate points, but I have written it for 1 mark in case this question might come for 2 marks also, so you can use this answer. Question number 5. A student investigated the effect of different concentrations of NaCl on osmosis in potatoes. The student's results are shown in table 1.1. Okay. Using the information in table 1.1, calculate the percentage change in the mass of potato cylinder in 0.8 mole per dm cube sodium chloride solution. Give your answer to 1 decimal place. Now I'll tell you just one formula here. Your formula should be here. Final minus initial over initial into 100. So this formula you have to take for percentage change. So your final is 0.8. Your initial is 1.14. Your answer has to be negative since your initial is more than final. So this will come up to a minus 28.1. Using the information in the table, explain why the difference in loss was greater in 0.8 mole per dm cube NaCl solution. Why do you think that the loss was more here in this case rather than the other ones? Because this solution had more steeper concentration gradient. So what I have written in my answer, let's see. 0.8 mole per dm cube solution has lower water potential than the other salt solution. More water molecules are lost due to steeper concentration gradient, which results in more decrease in mass. So there is more decrease in mass because more water molecules move out. Why did more water molecules move out? Because there was a steeper concentration gradient, and 0.8 mole per dm cube solution had a lower water potential than the others. Next, B. The diagram below shows the expected appearance of cell after it is immersed in distilled water for 30 minutes. Outline 3 differences that will take place in the cell after 30 minutes of immersion in distilled water. So, the differences that you can see in between them is vacuole size increases, the cell swells up, and the cell membrane and the cytoplasm are pushed against the cell wall. This is called as your turgor pressure. So, there is an increase in turgor pressure from the first to the second one. Describe how the process of active transport differs from diffusion. So either there can be like a question for active transport and diffusion or osmosis. So the points you can include here: active transport requires energy from aerobic respiration while diffusion does not. Same for osmosis also. Active transport requires carrier protein but diffusion does not. Same for osmosis. Active transport is from low concentration to high concentration and diffusion is from high to low. Same you can use for osmosis also. One more point which you can use for osmosis is active transport is for other molecules and not water, and osmosis is only for water molecule and not for any other molecule. Sixth question now. The ribcage and diaphragm are involved in breathing mechanism to ventilate the lungs. Figure 3.1 is a flowchart that shows the changes that take place when breathing in. Okay, so this question External, when you're breathing in, okay, so that's inspiration. So at that time, I'll just read the right side. Diaphragm muscle contracts, diaphragm is contracted, okay, at that time. Volume of the thorax increases, pressure of air in the lungs decreases, atmospheric pressure is higher than the air pressure in the lungs, and air moves inside the lungs. Air enters the alveoli, which are the site of gaseous exchange. So these are the points that we need to write related to inspiration. Okay, so this we had to complete it, but we have already completed. Coming to question number 7, marine conservationists are concerned that fish stocks in the sea are decreasing. Drastic measures will have to be taken up to stop the extinction of many fish species. Figure 5.1 shows a marine food web. Tuna are large carnivorous fish that are an important human food. Dolphins may be caught in fishermen's net and die. Okay, so this is the food web that you can see. State the names given to trophic level 1 and 3. Now please pay attention. The first trophic level here is of producers. So trophic level 1 is producers. Then primary consumer, secondary consumer, tertiary consumer, and quaternary consumer. So this is your quaternary consumer, this is tertiary consumer, this is your secondary consumer, and primary. So it is asking you about first and third. So this is producer, okay, and this is your about 3 is secondary consumer. Okay, so this is the answer I have written here. B part. Explain why it is more energy efficient for humans to eat herbivores fish rather than tuna. So it is asking you why it is more energy efficient to eat this fish rather than this fish. So you can just have a look at the answer what I have written. Energy transfer between the trophic level is inefficient. Energy is lost in respiration, excretion between the trophic level. You can write many other processes also. Herbivorous fish is more near to the producers and has more energy than tuna. So as we all know that energy transfer between the trophic level sharply decreases. It's not efficient, and that is why more energy is available at the bottom level near to the producer than the top ones. C part. Explain why it is necessary to conserve animals such as tuna and dolphin which are at trophic level 4. Why do you think that it is necessary to conserve any animal? Let's just read out the points. To prevent disruption of food chains, to maintain biodiversity, less food will be available for consumers and can cause extinction of species. See, if there is any imbalance in the food chain, it's going to lead to imbalance of the entire food web. So this will disrupt the entire food chain. It will reduce the biodiversity because organisms are getting extinct, and less food will be available to consumers, can cause extinction of species, reduce the number of herbivores. So this is how it is going to happen if any of the animals are going to be disturbed. D part. Many seas are polluted by non-biodegradable plastics. Suggest the likely effects of this pollutant on the marine environment. So how can you can say your plastics disturb the marine environment? Both is there, terrestrial and marine, in your syllabus. So this can get entangled around the fish and the other animals and cause suffocation. It can be eaten up and it can lead to death. So there is no space available for the food also. They will eat up and they will die because of that, because there is no space available for the food, and it does not break down naturally and releases toxic chemicals in the soil and harm the soil, microbes as well as plants, and can be mistaken as food by many other organisms. So they can also die because of lack of food. So all this you can include in this answer. So this basically sums up the entire paper. We have finished the entire predicted paper. In case you have any doubts, you can just let me know. I'm there to help you out, and do very well. All the very best for your exam. Again, I say the same thing. Be very calm and focused while reading the questions. You know everything. Just focusing bit in the questions, reading them properly will help you answer better. All the very best. Thank you so much.